received a few hours before 5-ALA, our compound, and then they were treated with sonodynamic therapy. The phase 0 component was that the patients only received treatment on half of the tumor. So patient got IV of the 5-ALA, but since this is a focused ultrasound, the patients all received treatment on half of the tumor, while the other half didn't receive any treatment. And then those patients, 4 days later, will have their plan already resection. Okay, and this is called a phase 0. The therapy really was not given with the idea of having a long-term effect on the patient, although, as was presented at the Society of Neuro-Oncology meeting, some of these patients did have a prolonged survival. But in our case, the objective of the study was to look at the tumor 4 days after they treated only half of the tumor. So 4 days later, Dr. Sinai went now to have the regular surgery, extirp or took out the whole tumor. And remember, we knew half of the tumor was treated with sonodynamic therapy, the other half wasn't. And when we look and did studies to allow us to see different, what we call biomarkers of cell death. So these are biochemical studies that we do on the tumor that indicates if death is already happening into the tumor. We found that there were the two more common markers that we know had to deal with cell death were statistically significant elevated on the half that was treated compared to the half that was not treated. So this is what the phase 0 told us, that we already very early, 4 days after the therapy, there was already biochemical changes on the half of the tumor due to the sonodynamic therapy. So in conclusion, each patient served as its own control. So half that was treated and the other half that was not treated. And we could show that difference in between the two halves and show that the part of the tumor that was treated already show signs of self-death compared to the untreated side. That was the phase 0. And the therapy was very well tolerated. At the same time that we gave that IV drug, we took samples of the patients to measure the pharmacokinetics, and we learned how to give this drug before the sonodynamic therapy and how it's eliminated by the body. So this combination of the data of initial biochemical efficacy, the safety of the procedure, and the pharmacokinetics of the drug that were given, then allows then to conclude that there was activity, safety, and a product that was stable. It was distributed in an expected way. And that's where we moved in now with the current study, or the two current studies, one in a pediatric tumor called the IPG, and again in recurrent glioblastoma patients. But now this was based on the activity and the drug doses that we saw at Dr. Sinai's place.
Right. So in this phase 0-2 study, you are testing if an approach works. You mentioned that half of the brain tumor received treatment, while the other side did not. Tell me a little bit more about this.
So remember, these patients already got the standard of care for the newly diagnosed patients. So now the patients completed, they have the initial therapy, the initial surgery, the initial radiation, the initial chemotherapy. Sometime later, patient comes for a checkup and has again the tumor. So these are recurrent tumors. So they were treated for the standard of care at the beginning, and they are going to go for surgery again. So that's when we came in. So the patients still received the standard in the sense that they had the surgery. But before the surgery, we did the sonodynamic therapy half and half. And when the tumor came out after the regular standard of care, the surgery the patient had planned, is when we saw the difference. So we did not compare to standard of therapy, what we evaluated in the same patient was the efficacy of the therapy in half of the tumor compared to the other half of the tumor. So there was no risk for the patient that we were giving something that was not proven, because at the end, the only proven therapy that this patient were planned to get was, again, another surgery, which just came before the surgery. This therapy took advantage that the tumor was coming out to look into the biochemical changes after we did the sonodynamic therapy.
In this 02 study, outcomes were already seen after a very short period of just 4 days. After the conclusion of the initial study, were you able to continue treating patients?
Correct. So as I mentioned, the phase 0 objective and phase 1 is really to look into phase 0 to look at biochemical responses, which we obtain, and phase 1 is safety and look at the distribution of the body, of the drug through the body. So in general, those patients are not set up to look for really long-term efficacy. But as I mentioned, there's already some presentation, although the publication hasn't come out yet, that there was some patients that have long-term responses. What we're doing now is doing a phase 1, phase 2 study, right, where we are confirming the safety and the drug distribution, the PK, in patients with GBM, with recurrent GBM. And then we're expanding to a phase 2. And that's when, in a phase 2, is when you look at efficacy and you have what they call endpoints of efficacy, like overall survival, how long did it take to the tumor to come back, if that's the case. So the objectives of the phase 0, phase 1, really, even though we like to know, and it's great to hear if a patient responded, that's not the objective of phase 0, phase 1. Phase 2, which is where we're conducting right now, it is where we are what we call powering the study. What does that mean? We need to have enough patients to show statistically that the response is caused by the therapy they were given and not by chance. And this is part of doing clinical trials, and that's where a phase 2 is. And eventually, in a phase 3, you will compare your therapy that looked positive in a phase 2 and then compare it to the standard of care. And again, you have to show a statistically significant difference in between the control or standard of care group and the experimental arm that has this new therapy. And that's when new therapies get approved by the FDA. So we are right now in that phase 2. And so far, we're starting. We have to wait to see the results of the study. But we already have a show in the IPG in children that we started treating for this terrible disease, that the safety is very good. We haven't seen any side effects on the children that have been treating with this. Again, the first 10 patients that were treated in Phoenix also didn't show any side effects.
Let's take a short break, and then we're back with Dr. Eli Benaim and Dr. Mark De Souza.
Sarcoma. Odds are you've never heard that word before. For the 40 people diagnosed with sarcoma every day, it is a life-changing word, because sarcoma is cancer. Through awareness, advocacy, and research, the Sarcoma Foundation of America is bringing hope to the families whose lives have been turned upside down by a cancer they had never heard of until diagnosis. Please join us in the fight to find the cure for sarcoma. For more information on the work of the Sarcoma Foundation of America, go to curesarcoma.org.
This is the Onco'Zine Brief with Peter Hofland.
And welcome back. In today's episode of the Onco'Zine Brief, I'm talking with Dr. Eli Benaim and Dr. Mark De Souza. I'm Peter Hofland, and this is the Onco'Zine Brief. Now, in these early studies, you're dealing with recurring cancers, right? And for these patients, it's not the first time that they are diagnosed with glioblastoma or any other form of brain cancer. Does that make a difference in terms of development of a particular approach?
So the issue is, in general, let me go back into GBM. So GBM in general, and many cancers where they recur, have very bad prognosis. That means that usually the results are worse than when they're newly diagnosed. And this is because the patient already have received a lot of therapies, had created resistance. The cancers are very smart. They create resistance through different mechanisms, including other chromosomal mutations or mutations of the tumor, and make the cells resistant to new therapies. So in general, happens many times that many tumors, they don't have many more things to offer after they recur. Some others, and this is when you hear patients in first line, that's when they are newly diagnosed, get certain therapy. Then when they recur, they may get a second line, and rarely there's some cancers that may get a third line that are already approved. In general, for second or third line, many times patients go into clinical trial. The way that the regulations are written is in a way that we need to protect the patients, correct? And there has to be a beneficence, has to be a benefit of the patient of getting an experimental therapy. So we need to be sure that we don't take away from the standard of care, that we don't give a suboptimal therapy with a patient when there's already a therapy that already has a benefit on the patient. So in general, in cancer, we treat patients in recurrence because there's not that many therapies available. So it is ethical to approach these patients and treat them with the new therapies. To treat a first line patient, you generally has to demonstrate in recurrent disease that there is activity to then take the risk of bring the therapy earlier with the chance that you may be given a suboptimal therapy compared to the standard of care. But, in our case, our therapy really, it's more of a neurosurgical technique without the scalpel, because we are giving a nutrient to the body and then coming out and doing surgery without opening the brain, without giving side effect, and then potentially, having a great benefit to the patient. So the development plan is really to try to have enough data soon in these recurrent diseases to bring this therapy earlier in the patients before they get radiation, before they get chemotherapy, even before they have to have major surgeries, and try to see if we can treat these patients, take control of the tumor, eradicate the tumor, and before or not give any more therapy after. So that's basically the development plan. And one more thing about this therapy, it is since we're giving a nutrient to the body that is only taken or is mostly taken by the tumor cells, which they cannot survive without it, then the chances of create resistance are very low. While other chemotherapies or other therapies will create resistance when they create new mutations. For us, it is not. It doesn't affect our therapy if the patient has a mutation or not, because our therapy treats patients that are rapidly divided by the cancer cells. So any cell that reproduces very quickly is going to require a lot of 5-ALA and is going to be susceptible to sonodynamic therapy.
To clarify, patients are receiving a particular drug which is followed by a radiologic or ultrasound approach. Is that correct?
Yeah, that is the case. Since the cells cannot keep producing a protein is called heme that carries oxygen inside the cell, it accumulates this, what we call protoporphyrin 9 or PP9. So it's the malignant cells by itself creates the cell the product that is susceptible to the sonodynamic therapy, while the normal